All right. Good morning, everybody. Today is Thursday, October 5th, 2017. And this is Before the First Cup with Jules here on UCY.TV. Happy Thursday, everyone. It is a partly sunny 60 degrees and a pleasant fall day here in upstate New York. So where do we leave off yesterday? We titled the show CIA, Harvard, IARPA, Brain Mapping, MK Ultra, and Mind Control. And we dug into the cozy relationship that the alphabet agencies have had with Harvard over the years, starting back in the days of the MK Ultra program, but even getting through to recently with IARPA, which is the intelligence agency's version of DARPA, and Harvard and Georgie Porgy Church for barcoding neurons and brain mapping to be able to understand how flipping switches interact with the living organism. Well, today we're going to look at flipping switches, but it's not going to be in the brain or with neurons. It's going to be with CRISPR. Again, I ran across quite a number of stories over the last day or two that really made me raise an eyebrow, and I wanted to bring it to your attention. This one was actually from 2015. We had talked about utilizing CRISPR for genetic modification and some of the things that DARPA had been looking at with the Insect Allies program to be able to infect an insect with a virus or a bacteria, and then within that virus or bacteria to put a payload of CRISPR, which is the immune system of an E. coli bacteria, inside of the virus of bacteria. And then when the insect goes to eat the plant or the crop, it infects the plant or the crop with its spittle with whatever the contagion is that it is carrying as a Trojan horse payload. And that then releases the CRISPR technology into the plant to go genetically modify that plant while it is live and still growing. Well, they have been working on very similar delivery vectors for CRISPR in humans. New Cas9 molecule points the way to viral delivery of CRISPR systems. Yes, CRISPR delivered via viruses. Startup biotechs and major pharmaceutical companies alike are working overtime to apply the recently discovered CRISPR genome editing technology to treating or even curing human diseases derived from a natural defense mechanism of bacteria. The CRISPR system uses a class of proteins called Cas9 to engineer precise cuts in the cellular DNA, making it possible for scientists to flexibly delete or insert genes anywhere in a genome. There are obstacles to using CRISPR in living organisms, however, and perhaps the biggest problem is delivering Cas9 to the cells in the body. And the most popular vector for gene therapies, in which now DNA sequences are inserted into the human genome to combat the effects of disease-causing mutations, has long been the adeno-associated virus, more affectionately known as AAV, which can deliver a genetic payload to a single, well-defined region of the genome without noticeable side effects. However, the AAV vectors have been largely unavailable to CRISPR scientists because these viruses can only carry payloads up to certain sizes, and a standard Cas9 system, including the Cas9 molecule itself and multiple guide RNA sequences to direct the Cas9 to its DNA target, come in over the size limit. Today, April 1st, 2015, a paper in Nature demonstrates a way around this limitation, successfully engineering genetic changes in mice using an AAV-delivered CRISPR system. The work comes from the lab of Fang Zhang, and we're going to look at Fang Zhang of the Broad Institute of MIT and where? Oh, Harvard, yes, who first demonstrated that CRISPR genome editing could be performed in mammalian cells in 2013. Almost all scientists using CRISPR today choose a Cas9 molecule derived from the bacterium, and this one is the Streptococcus pyogenes. We've also read about the E. coli. And the reason is the Streptococcus pyogenes has been used as a standard because it is the only Cas9 that was very reliable in terms of efficiency and also has a broad range of targeting, explains Li Kong. co-author of the new paper, and a postdoctoral researcher at the Broad Institute. The efficiency refers to how reliably the Cas9 molecule cuts both strands of DNA at the targeted site, while the targeted range refers to how many potential regions of the genome of the molecule it can interact with. Now, let's stop for a minute because I pulled up a document looking at what this adenovirus is. An adenovirus, a DNA virus, which was first isolated in the 1950s in the adenoid tissue, delivers cell cultures, hence the name. These primary cell cultures were often noted to spontaneously degenerate over time, and an adenovirus are known to be common causes of asymptomatic respiratory tract infections that produce in vitro cytolysis in these tissues. An extremely hardy virus, adenovirus is ubiquitous in humans and in animal populations. Ding, ding. Survives long periods outside of the host and is endemic throughout the year. So, look, we got it 12 months out of the year. Survives a really long time outside of the host, and it is ubiquitous in humans and animal population. What a great way to create a bioweapon or some sort of future pandemic. Possessing 52 stereotypes, the adenovirus is recognized as an etiologic agent of various diverse syndromes. It is transmitted via direct inoculation to the conjunctiva, which is the eye, the fecal oral route, as aerosolized droplets or exposure to infected tissue or blood. The virus is capable of infecting multiple organ systems. However, most infections are asymptomatic. Well, how great is that? Because you don't even know you might have it. Adenovirus is often cultured from the pharynx or the stool of asymptomatic children, and most adults have measurable titers of anti-adenovirus antibodies, implying prior infection. Adenovirus is known to be achinogenic in rodents, but not in humans. Adenovirus has been associated with both sporadic and epidemic disease, and with regards to infections among military recruits who are routinely immunized against types 4 and type 7 from 1971 until the cessation of the vaccine production in 1996. Adenovirus became a significant cause of economic cost and morbidity in this setting, and the live oral vaccine against adenovirus types 4 and 7 was approved for use in the population by the U.S. Food and Drug Administration in 2011, and subsequent incidences of acute respiratory disease declined. Of interest is the role of adenovirus as vectors in vaccination and in gene therapy. Adenoviruses can infect various cells, both proliferating and quiescent, and thus hold promise of targeting many different tissues and disease cell lines. The genome of the adenovirus is well known and can be modified with relative ease to induce lysis or cytotoxicity of a specific cell line without affecting others. The virus itself can be engineered to remove its replicative capacity by removing essential genes, and additionally, specific genes can be inserted into adenovirus that then can repair defective metabolic, enzymatic, or synthetic pathways in the host. Suicide gene systems that convert non-toxic systematic delivery pro-drugs to active chemotherapeutic agents have been delivered via adenovirus vectors directly into cancer cells. However, the greatest challenge in viral gene therapy, as might be expected, is the human response to the vector itself. The complex mechanisms by which viral vectors may be incorporated into gene therapy and the rapid growth in this field put further discussion beyond the scope of this text. All right, so now let's take a look at who the Broad Institute is, if it'll load, and who they're involved with. The Broad Institute announces DARPA awards them a $32 million contract September 24, 2015 to MIT Broad Institute Foundry to bolster DNA design and manufacturing. The facility at the Broad Institute of MIT and Harvard that aims to achieve the full potential of engineering biology has received a 5-year, $32 million contract from the Defense Advanced Research Project Agency, more affectionately known as DARPA. Cambridge, Massachusetts, a facility at the Broad Institute of MIT and Harvard, aims to achieve the full potential of engineering biology with a 5-year, $32 million contract from DARPA. The foundry, started by MIT biological engineering professor Christopher Voigt and Broad Technology Labs director Robert Nicol, is the result of a partnership between Broad Technology Labs and the Synthetic Biology Center of MIT, of which Voigt is a co-director. The foundry enables the rapid design, testing, and fabrication of large sequences of genetic information so they can be assembled like building blocks for a myriad of medical, industrial, and agricultural applications. Society relies on many products from the natural world that have intricate material and chemical structures, from chemicals such as antibiotics to materials like wood, says Voigt. We have been limited in our ability to program living cells to redesign these products. For example, to program living cells to create materials as intricate as wood or seashells, but with new properties. And rather, products from synthetic biology have been limited to small, simple organic molecules. I want to change the scale of genetic engineering to access anything that biology can do. Now, if you remember the DARPA lady talking about being able to terraform Mars in the future by splicing and dicing organisms from across the board of life to make whatever type of organism they need. Let's talk about now Feng Zhang. Feng Zhang is the one who worked on some of the adenovirus infection of organisms with CRISPR. Feng Zhang, there was a brand new story about him, September 28, 2017. Feng Zhang joins Society for Science and the Public Board of Trustees. The Society for Science and the public is excited to share the noted scientist Feng Zhang has joined the Society's Board of Trustees. Zhang is a core member of the Broad Institute of MIT and Harvard, an investigator at the McGovern Institute for Brain Research, and the James and Patricia Poitras Professor of Neuroscience at MIT, an associate professor in the Department of Brain and Cognitive Sciences and Biological Engineering at MIT, and the New York Stem Cell Foundation Robertson Investigator. Zhang is also an alumnus of two of Society's world-class science competitions, the 1998 and 1999 International Science and Engineering Fair, as well as the 2000 Science Talent Search. Feng is an innovator whose breakthrough ideas are changing the face of bioscience, says H. Robert Horvitz, chairman of the Society's Board of Trustees. I am confident he will add great value to our Board of Trustees, and the Society for Science and the public will benefit from his experience and counsel. As an alumnus of both of our high school science competitions, Feng will be an informed and insightful point of view to the board and their deliberations. Let's take a look at what Feng actually has listed. This is out of MIT, and this is his main page directory for Feng Zhang, PhD, core member, Broad Institute of MIT and Harvard, investigator at McGovern Institute for Brain Research, MIT, associate professor of the Department of Brain and Cognitive Sciences and Biological Engineering at MIT, and New York Stem Cell Foundation Robertson Investigator. Research at a glance, synthetic biology, development of technologies for genome and epigenome editing, neural circuit engineering, optogenetics, stem cell engineering, and gene delivery. Feng Zhang attended Harvard University, as an undergraduate, where he earned an A.B. in chemistry and physics in 2004. And then he transitioned to Stanford for his PhD in chemistry and biophysics, which he obtained in 2009, while working with Carl Deseroth's research group for his work pioneering optogenetics in conjunction with Ed Boyden. After a postdoctorate in where? Oh, Georgie Porgy Church's lab. He moved to MIT in January of 2011, where he's now both an assistant professor of biological engineering and a core institute member of the Broad Institute of MIT and Harvard. Well, this sounds just like the kind of guy that maybe IARPA and DARPA want to get their hands on. Fang Zhang has received a number of honors, including the National Institute of Health Pioneer Award and the Pearl UNC Neuroscience Prize, along with Ed Boyden and thesis advisor Desarath. He is additionally a Searle Scholar. His research is about the mammalian brain and how it poses a formidable challenge to the study and the treatment of neuropsychiatric diseases, owing to a complex interaction of genetic, epigenetic, and circuit-level mechanisms underlying pathogenesis. Technologies that facilitate functional dissection of distinct brain circuits are necessary for systematic identification of disease origin and therapy. The Zhang Group is developing and applying molecular and optical technology for probing brain function in health and disease, and the lab hopes that these new applications will improve our understanding and our treatment of brain diseases. And don't forget that he's also working on finding out how to get CRISPR into the body by infecting you with an adenoid virus. Now listen to this. This is out of the National Institute of Health. Method for dual viral vector mediated CRISPR-Cas9 gene disruption in primary human endothelial cells. Human endothelial cells are widely used to study mechanisms of angiogenesis, inflammation, and endothelial permeability, and that would be to the bloodstream through the vessels. Targeted gene disruptions induced by CRISPR gene editing is potentially an important tool for definitively establishing the functional roles of individual genes in endothelial cells. We showed that code delivery of adenovirus-encoded EGFP tagged Cas9 and a lentivirus encoded a single guide RNA in a primary human lung microvascular endothelial cell, and it disrupted the expression of TIE2 genes and proteins. TIE2 disruption increases the basal endothelial permeability, so they were able, through the lung, to get this adenovirus in, and then this adenovirus affected the interior of the blood vessels to make them more permeable, to open them up to the bloodstream, to allow things in. And by disrupting these genes and proteins, they increased the permeability, and the permeability recovery following injury was prevented by the inflammatory stimulus of thrombin, and this is by their gene therapy. Thus, gene deletion via viral code delivery of CRISPR in primary humans' endothelial cells provides a novel platform to investigate signaling mechanisms of normal and perturbed endothelial cell function without the need for clonal expansion. So in other words, they can get CRISPR in through your lungs utilizing adenoviruses, and once they get them into your lungs, they disrupt the endothelial cells that are protecting your bloodstream from infection or infiltration to be able to get things inside of your bloodstream. Now that sounds exactly like what they would want to do. Now, remember we took a look at how some of the alphabet agencies, including DARPA, were trying to figure out how to protect humans from being a victim of a biological attack utilizing CRISPR, which has been deemed a WMD by Clapper. So there was a program that came out titled Safe Genes, and this was sponsored by DARPA. The Safe Gene program aims to deliver novel biological capabilities to facilitate the safe and expedient pursuit of advanced genome editing applications while also providing the tools and methodologies to mitigate the risk of unintentional consequences or intentional misuse of these technologies. There are 3 primary technical focus areas within the Safe Gene program to help bridge the gap between the current technological landscape and the future transformative applications of genome editing tools and engineered organisms. And remember, we read a few different times now how the security has been very lax in these laboratories to boot. First, and the technology has been democratized by AddGene across the globe. First, researchers will design and develop the genetic circuitry and genome editing machinery for robust spatial, temporal, and reversible control of genome editing activity in living organisms. The second technical focus area will develop small molecules and or molecular strategies to provide prophylactic and treatment solutions that prevent or limit genome editing activity and protect the genome integrity of organisms and populations. Finally, researchers will develop quote-unquote genetic remediation strategies that eliminate unwanted genetically engineered genes from a broad range of complex populations and environmental contexts to restore systems to functional and genetic baseline states. Everyone's going to have to step right up and get their genome put into a database. So just in case there's ever an accidental on-purpose release of these technologies into the population, we can set you back to baseline. It's just insane. Overall, the program aims to create a layered, modular, and adaptable solution to set access translational applications of genome editing technologies while protecting against intentional or accidental misuse of these tools. Now, Wayne, I just found a story about who actually won some of these grants. Hold on, let's get it to load. Safe Genes, and this was a story from July 25th, 2017. Safe Genes Award to Massachusetts General Team aims to improve gene editing technologies. The Massachusetts General Hospital-based research team is one of 7 nationwide receiving contracts under the DARPA Safe Genes program, which is designed to improve the safety of gene drives, which Billy Gates is doubling down, investing them in, techniques that can ensure a beneficial gene variant that spreads throughout a population of organisms much more rapidly than through normal inheritance. And by the way, Massachusetts General Hospital is the teaching hospital that is connected to Harvard University. Ding! The group, led by J. Keith Zhuang, MD and PhD, Associate Chief for the Research of the Massachusetts General Hospital Department of Pathology, also works toward developing better ways to detect and prevent off-target mutations induced by CRISPR-Cas9 gene editing nucleases. The CRISPR-Cas9 technology utilizes elements found in bacterial immune systems, including DNA-cutting nucleases, that enable it to induce breaks at specific gene sites and then insert new genetic material at desired locations in the human genome. Ding, ding. In 2013, Zhuang's team at Massachusetts General was the first to report that nucleases could also have unwanted off-target effects in human cell lines. And we read that story. There was over 1,500 off-target effects they discovered in the one experiment with treating blindness in rats, I believe, and inherited blindness, raising an important issue to be evaluated prior to therapeutic applications. Since then, his team has developed a way of detecting unwanted CRISPR-Cas9-induced DNA breaks in engineered cells and created a high-fidelity nuclease that induced no unwanted mutations that could be detected by that method. Although we've made a lot of progress over the last 3 or 4 years, we still want to have even more sensitive and robust ways of finding these off-target effects, Zhuang says, and that is important not only for developing safe therapeutics, but also for gene drives developed to propagate a genetic change through an entire population of, for example, mosquitoes, and developing The development of better assays should, in turn, help us engineer even more precise forms of CRISPR-Cas9 nucleases. Zhuang's Safe Genes team is receiving more than $10 million from DARPA for the project and includes investigators at the Imperial College London, which is where Oxitec came out of, which is where genetically modified mosquitoes came from, as well as North Carolina State University, Boston University, and Polo GGB, which is a nonprofit research organization based in Italy. Our focus will be developing even more sensitive methods of measuring and controlling on-target gene editing activity and detecting and limiting off-target activity. They also hope to develop better means of controlling gene drives, including a more precise way of regulating the expression and the activity of CRISPR-Cas9 and other gene drive elements. Efforts at the Massachusetts General will be spearheaded by Vikram Patnaik, MD and PhD, also of Massachusetts General Pathology, and an instructor of pathology at where? Oh, Harvard Medical School. A professor of pathology at Harvard Medical School and the Desmond and Anne Heathwood Massachusetts General Research Scholar, Zhuang says, "We are delighted that DARPA sees the need for this and is willing to fund this type of research, which is a good fit with our continuing interest in pursuing these important questions. Our primary focus is improving the safety of these tools, of course it is, and our findings should lead to safer therapeutics and gene drives, as well as better research and reagents." The other 6 safe gene teams are led by Amit Chandri, PhD of the Broad Institute, Ding, and the Brigham and Women's Hospital, as well as Georgie Porgy Church, PhD at Harvard Medical School, Kevin Asvelt, a PhD at Massachusetts Institute of Technology, John Goodwin, PhD of North Carolina State University, Jennifer Doudna, PhD of University of California, Berkeley, and Omar Abakarai, PhD at the University of California, Riverside. Massachusetts General Hospital, founded in 1811, is the original and largest teaching hospital of Harvard Medical School. The Massachusetts General Research Institute conducts the largest hospital-based research program in the nation. Yeah, of course it does. And an annual research budget of more than $800 million in major research centers for HIV/AIDS, cardiovascular research, cancer, computational and integrative biology, cutaneous biology, genomic medicine, and medical imaging, as well as neurodegenerative disorders, regenerative medicine, reproductive biology, systems biology, photomedicine, and transplantation biology as well. Massachusetts General topped the 2015 Nature Index list of healthcare organizations publishing in leading scientific journals and earned a prestigious 2015 Foster G. McGraw Prize for Excellence in Community Service. In August 2016, Massachusetts General was once again named to the honor roll in the U.S. News and World Report list of America's best hospitals. And I bet this is where they send all of the senators and upper level politicians when they need treatment. All right. So DARPA announced, let's see if there's a date, July 23rd, 2017, their Safe Genes program, and put $65 million in funding behind it. The 7 teams, blah, blah, blah. The awarded funding by the program includes, of course, Georgie Porgy Church at Harvard Medical School, which will use the DARPA funding to develop methods to protect genomes by detecting, preventing, and ultimately reversing mutations that may arise from exposure to, things such as radiation, he says. Church and his team are planning on creating new computational molecular tools to enable the development of precise gene editors that can distinguish between highly similar gene sequences. His team also plans to screen the effectiveness of drugs that inhibit gene editing activity. Church believes that the gene editing tools are even more accurate and easier to use than CRISPR are possible in the near future. And guys, remember, Georgie Porgy Church is working with IARPA as well as DARPA. IARPA with the barcoding Apollo project of the brain. And there is also a project that is going on right now with IARPA for the detection of genome editing. Ding. The Intelligence Advanced Research Projects Agency, more affectionately known as IARPA, is seeking information on potential tools and methods to detect organisms who have been modified using genome editing techniques. This request for information is issued solely for information gathering and planning purposes, and this was put out January 23rd, 2017. Responses were due March 3rd, 2017, so that was this year. Solely for information gathering and planning purposes, it does not constitute a formal solicitation for proposals. The following sections of this announcement contain details of the scope of technical efforts of interest, along with instructions for the submission of responses. Recent breakthroughs in genome editing, including CRISPR-Cas9, remember this is the intelligence agencies, DARPA, are enabling significant advances in life sciences. Beneficial impacts are expected to be wide-ranging, from creating novel medical treatments to the development of crops that are more nutritious and able to grow in harsh environments. Wink, we're going to gene edit the world. In addition to gene drive technology, may provide opportunities to prevent diseases in crops, remove invasive species, and stop the spread of malaria, which Billy Gates is investing heavily in, and other vector-borne diseases. While genome editing technology will enable new biological breakthroughs, the unintentional or deliberate misuse of gene editing technologies may have adverse economic, health, and national security implications. So IARPA is interested in tools and methods that can be used to detect evidence of genome editing in biological organisms, including viruses. They're going to be scanning each and every one of us. Oh, you have been infected. Time to get your Georgie Porgy Church solution to turn back the clock, take you back to baseline. The fundamental components of many genome editing tools are found in nature, so detection requires an ability to distinguish engineered organisms from naturally occurring background. Detection capabilities will help guard against unintended outcomes of genome editing and expedite appropriate mitigation responses for these unapproved releases of edited organisms. I'm sorry about that, Johnny, but your DNA sequence is unapproved. We're going to have to set you back to where you belong. Within this topic, area of interests include, number one, research aimed at identifying direct or indirect signatures of genome editing in biological organisms. Number two, associated with edited microorganisms, plants, and animals, especially those of economic importance, which would be crops, livestock, and pollinators, or those that serve as infectious disease reservoirs, such as insects. Number three, research efforts, including those in genomics, proteomics, metabolomics, and that can better distinguish organisms that are engineered from those that occur naturally. Number four, scalable and or high throughput approaches for detection of organisms that, if altered, have a health or economic implication, including ecological and agricultural. Number five, methods to estimate the longitudinal stability of modifications once moved from a laboratory setting to the environment, and factors that determine if a modification is entirely lost or if markers of the modification still exist, so we can hide it and no one will be able to tell. I think it's 6, methods for predicting and anticipating environmental effects resulting from a release of an engineered organism. And number 7, determination of provenance of gene editing tools and techniques. Responses to this RFI should answer any or all of the following questions. 1, what are the computational and laboratory-based tools or methods that are currently available for detection of genome editing? How accurate with regards to sensitivity and specificity are these methods, and how do they compare to each other? What are the limitations of these approaches, and do they vary depending upon species? What other types of methods could be developed or repurposed that have a potential to detect gene-edited organisms? Are there methods or approaches for certain detection that are agnostic to the type of genome editing technology used to make the modification? What tools can be developed that integrate data from new and or existing databases for analysis or genetically altered organisms? What are your time frames, and what technological advances will be required to shorten the time? What is the data-driven modeling approach, if any, that can be developed or leveraged to assist with the detection? And what opportunities currently exist or can be developed for persistent detection of organisms, that means 24/7, that have been modified using genome editing techniques? Are there existing mechanisms in use by government, academia, or industry that can be leveraged as a detection capability? For example, can existing screening measures for mosquito-borne diseases also be used to investigate incongruent signatures? And if so, what are they and how can they be implemented, and what are their associated challenges? Well, talking about scanning for genetically modified organisms, let's take a look at some of the newest advances in CRISPR to come out over the last couple days. October 4th, 2017, CRISPR nanoparticles repair Duchenne muscular dystrophy gene. Scientists have developed CRISPR Gold, which is a gold nanoparticle technology for delivering CRISPR-Cas9 gene editing to cells that, when tested with the MDX mouse model of Duchenne's muscular dystrophy, which is DMD for short, repaired the faulty DMD gene, leading to improved strength and agility and reduced fibrosis. Professor Nurin Murthy, PhD of the University of California, Berkeley, which is where Jennifer Doudna is, led the experiment of the CRISPR Gold platform, suggesting that human clinical gene editing trials using the system could feasibly start within the next few years. CRISPR Gold uses gold nanoparticles to encapsulate all of the elements needed for CRISPR-Cas9 gene editing and deliver them directly to cells. So there are two types of CRISPR-Cas9 in the human body. There is virus vector and non-virus vector. This is considered non-virus vector because it is encapsulating them in gold nanoparticles and then injecting them into the muscular tissue to get them inside of the body. CRISPR Gold uses gold nanoparticles to encapsulate the elements needed for CRISPR-Cas9 gene editing and delivers them directly to cells. Initial tests in mice achieved levels of gene correction that could demonstrate significant clinical benefits if they can be reproduced in humans. Professor Murthy told GEN, which is this Genetic Engineering and Biotechnology News, we think a clinical trial for therapeutic gene editing with CRISPR Gold and its derivatives could happen within the next 5 years. However, he added, it is not clear whether initial trials will be in DMD or another disease. Duchenne's muscular dystrophy is very compelling first clinical applications of the CRISPR Gold technology. However, there are several others, and it is still too early to say what the first clinical application will be. Well, let's take a look here, because there is another CRISPR story, October 3rd of 2017. Researchers have used CRISPR to prevent glaucoma. Oh, this isn't an inherited blindness. This is a genetic form of a disease that causes blindness, raising hope of preventing glaucoma, one of the most common causes of irreversible blindness. Researchers have used a gene editing method, CRISPR-Cas9, to disrupt a mutant gene that is responsible for some forms of the disease. The study, published in the journal Proceedings of the National Academy of Sciences, involved the elimination of the mutated mycocillin protein from a mouse model of human glaucoma and cultured human cells throughout the use of CRISPR-Cas9, which can alter DNA sequences and gene function. Mutations in myocillin are implicated in juvenile and adult onset primary open-angle glaucoma. And in their experiments, researchers found that removing a mutated protein by disrupting the mutant mycocillin results in lower intraocular pressure, which in turn prevents glaucoma from damage to the eye. As scientists don't want to just discover a disease gene, they want to understand what the gene does, and in this case have a better understanding of glaucoma so that it can be more effectively treated. No one knows what this gene does except that it's a mutant form, and its mutant form causes glaucoma. Glaucoma is a common vision and neurodegenerative disorder that affects 3 to 5% of people over the age of 40. An early precursor to the most common form of this disease is known as primary open-angle glaucoma, and it is high intraocular pressure, fluid pressure inside the eye that damages the retinal ganglion axons at the optic nerve that leads to the death of retinal ganglion cells that carry the visual signals to the brain, which then can cause blindness. So look at that. They want to cure muscular dystrophy as well as glaucoma, editing live humans. Out of New Scientist magazine, October 2nd, 2017, we are nearly ready to use CRISPR to target far more diseases. Well, let's get excited now. The race is on to edit DNA in our body to fight or prevent disease. Promising results from animal studies targeting the liver, the muscles, and the brain suggest that CRISPR genome editing, which can get inside of your body utilizing an adenovirus through the lung endothelial cells and into your bloodstream, could revolutionize medicine, allowing us to treat or even cure a huge range of disorders. The CRISPR genome editing method was only developed in 2012, but don't get excited because it's proving so powerful and effective that around 20 trials in humans have already begun or will begin soon. Almost all of these involve removing cells from an individual's body, editing their DNA, and then putting them back into the body. This approach has an immense promise. For instance, it is being used to alter immune cells to make them better at killing cancer. It is relatively easy to remove immune cells or blood stem cells, edit them, and then return them to the body, but this is impossible with most bodily tissues. So, of course, we're looking at other ways to get CRISPR into the body. So editing cells inside of the body would allow us to treat far more conditions, from genetic disorders to high cholesterol, and it would also be cheaper than growing and editing cells outside of the body. What diseases could be treated this way? Well, absolutely everything, says Irna Conboy at the University of California, Berkeley. Liver disease. A big challenge is delivering CRISPR machinery to tissues inside the body. Edited genes with CRISPR requires at least two components: the protein that cuts DNA and the piece of RNA that guides it to the precise DNA site to make the cut. Proteins and RNA are enormous molecules compared with conventional drugs, but we found out they just got around that. And it's hard to get them inside of cells, and they usually don't survive in the bloodstream either. Of course, they can put them inside of an adenovirus and then have that affect the permeability of the endothelial cells to get it into the bloodstream. But biologists have been working on delivering big molecules to cells for decades and are now adapting various methods for genome editing. Intertalia Therapeutics of Cambridge, Massachusetts, again, for instance, is using fatty particles or lipid cells to deliver CRISPR components to livers. One aim is to treat a rare genetic disease called transthyretin amyloidosis, which causes an excessive production of TTR proteins. And last week, the company reported that it managed to disable the TTR gene in the livers of mice, reducing the level of proteins by 97% with no signs of any ill effects. Intellia is also working for a cure on hepatitis B, which infects 250 million people worldwide. The virus can be difficult to eliminate because viral DNA can linger in liver cells, while CRISPR can destroy this DNA. And the liver is the easiest organ to deliver fatty particles because it filters the blood. I also remember reading very early on in CRISPR that the liver is where a lot of nanoparticles tend to accumulate as well. In some of these therapies, anything injected into the blood supply is likely to reach the liver. Conboy's team has managed something more difficult. They have treated the muscle-wasting disease, muscular dystrophy, in mice by injecting gold nanoparticles carrying the CRISPR components directly into the muscle. What is more, Conboy's team actually fixed the faulty gene rather than simply disabling it, as Intertalia did. Repairing genes is much harder than disabling them. It only works in 5% of muscle cells, but that was enough to boost muscle strength. And Conboy thinks the proportion of repaired cells could be increased by repeated injections of the gold nanoparticles, although her group has not yet shown this. I think this delivery method is fantastic for certain applications, says Jeffrey Chamberlain of the University of Washington. However, to treat disorders such as muscular dystrophy, muscles all around the body, including the heart, need to be edited. They cannot be injected directly. So the group hopes to find a way to get the gold nanoparticles into tissues throughout the body. And some of the stuff that we've read in the past, you can get nanoparticles across that blood-brain barrier by utilizing ultrasounds. Maybe they should check that out. Chamberlain has been able to edit tissues all over the body, though, utilizing viruses. Hey, earlier this year, his team successfully treated muscular dystrophy in mice by injecting them with what? Oh, an adeno-associated virus carrying DNA coding for CRISPR components. So there we go, guys. They are using this adenovirus to get CRISPR inside of living organisms. But there's also a big safety issue with viral delivery. In the cutting protein that is added to cells, as in the gold nanoparticle method, it soon breaks down, but the viral method involves inserting the DNA for the protein into cells instead, which means that the cutting protein keeps being made for weeks after the treatment. This then greatly increases the chance of DNA being cut in the wrong place, says Chamberlain. Already, though, Nicole Deglan of Lausanne University Hospital in Switzerland and colleagues had developed a way to prevent the cutting protein lingering for too long when the viruses are used for CRISPR delivery. Their kamikaze CRISPR system not only disables the target gene, but also disables the gene for cutting protein after a short delay. Last month, her team showed that this system reduces off-target effects in mice and the gene that causes Huntington disease in their experiment. And what's more, they managed to disable the gene in 65% of cells in the key area of the animal's brains. I was amazed to see how efficient it was, says Deglan. Another range of methods for delivering CRISPR is showing promise, suggesting the technique's potential for treating some of our most difficult diseases could soon be realized. It is impossible to say how soon trials in humans will begin, but the field is advancing so rapidly that it may not be that long. It's going faster and faster, says Deglan. So put on a seatbelt, people, because CRISPR-Cas9 is coming for you. And there is one more treatment that I found. The End of Snoring, application of CRISPR-Cas9 genome editing for sleep disorders. Oh no, June 28th, 2017. Where does it start? I have the abstract, but hold the phone. This PDF isn't loading. CRISPR, adaptive immunity in some bacteria, blah, 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 blah. Come on, where does it start? Oh, no. Page 2, 3, and 4 is all blurred out. All right, hold on. We're going to see. We are unable to authorize full PDF access to this article at this time. Click here to pay $240 for it on the publisher's site. Well, sorry, I'm not going to do that. But they want a CRISPR to be able to cure snoring. They said that some snoring is caused by a genetic anomaly. Guys, this is where this slope is getting very scary. They're going to get this into people, and then they are going to use it to cure everything that ails you. Now, this is out of an Indian publication, the Economic Times, indiatimes.com. My research aims at making medical care affordable for the common man. We are working on CRISPR technology, and this is another Mad Shore Off Science Lab. India is one of our Mad Science Lab testing grounds. And we are working on CRISPR technology, the gene which is seen, which you can even regenerate your little finger grown on your own heart, which means you don't need any immunosuppression, blood grouping, or matching. Frontier Lifeline is one of my latest additions. I decided that I must have a hospital where it's not only for patients, it should have a lot of research activities as well. In an interview with Dr. K M Cherian, Chairman and CEO of Frontier Lifeline Hospital in Chennai, Dr. Cherian talks about the need for upscale research activities to make medical care affordable for the common man. I've decided that I must have a hospital where it's not only for patients, but it should have a lot of research activities. My main research has been mainly to make medical care affordable for the common man, which means you need indigenization, which I don't even know what that word is, and medical research. Having a small place in the city is not going to help, so we decided that we needed to have a land outside of Chennai City. We built up a 5,000 square foot research facility, and it includes all the faculties. We have been lucky to have enough collaboration from different universities, not only from this country, but from outside. We are working on CRISPR technology, the gene which is seen, from which you can even regenerate your little finger and grow your own heart cells, which means that you don't need any immunosuppression drugs, blood grouping, or matching. None of those, this is a horrible translation. None of these things you can grow your own organs, but stem cells you can. But it may not happen today, but CRISPR technology has proved, and I am glad, I can't read any of this. So it's going to be for the common man to cure everyone, even in India. But as this technology is spinning out everywhere, we have a company called Odin.com who puts out a DIY human CRISPR guide. This is by no means meant to be a comprehensive guide, and instead meant to show people the ease at which CRISPR-Cas9 can be used to modify the adult human genome. And you can buy kits too through this Odin. CRISPR-Cas9 is a system with a terrible name that, contrary to popular belief, does not actually do any genetic engineering and modification of the DNA bases directly. Instead, it uses a system to trick that has been well known in the genetic design for many years. It cuts DNA. And yep, that's all Cas9 does, is cuts DNA. And you remember in an interview we listened to with Georgie Porgy Church way back when, he had said that in the very beginning, CRISPR-Cas9 was a tool of destruction, not a tool of prevention, because all it did was destroy DNA. So when DNA is damaged or cut, most of all organisms start to do DNA repair, and this can end in one of three ways. The DNA is repaired perfectly and everything is okay. The DNA is repaired, but some mistakes happen, leading to problems translating the gene into protein because the frame shifts or it mutates. And number three, the DNA is repaired using a template artificially supplied that results in a completely new sequence, allowing them to gene edit. And when the CRISPR people aim to make use of two or three, but most of the time when people talk about CRISPR, they mean that they just number three, which is not always possible. Most all CRISPR systems are composed of 2 to 3 components: a Cas9 protein, which cuts the DNA, a cRNA, and RNA, which synthetically combined with what is called an RNA guide, but also is called an sgRNA, and then the template for repair if doing homology-directed repair. So which Cas9 should I use? And he goes through designing your RNA guide and tells you all how to do it. But back in February 9th, 2016, James Clapper, the U.S. Director of National Intelligence, came out to the U.S. intelligence community and said that the top threat to the United States is gene editing technology as a WMD. That is according to James Clapper, U.S. Director of National Intelligence, who on Tuesday of 2016 said at the annual Worldwide Threat Assessment Report of U.S. Intelligence Community, gene editing has been added to a list of threats posed as weapons of mass destruction and proliferation. And remember, if they can do insect allies, which DARPA is looking at doing, why couldn't a rogue nation or a rogue element genetically modify an insect to come in and start biting humans like fleas, ship them in on a cargo vessel, and then start overtaking our forests and our wildlife to be able to spread the disease vector. Remember, insects as disease vector are as old as sin, and they've been looking at that for a very long time. Gene editing refers to several novel ways to alter DNA inside of living cells, and the most popular method, CRISPR, has been revolutionizing scientific research, leading to novel animals and crops, and is likely to power a new generation of gene treatments for serious diseases, as we have just heard. It's gene editing's relative ease of use, even DIY kits, that worries the U.S. intelligence community. And given the broad distribution, the low cost, and the accelerated pace of development, and how we are funding a Department of Defense initiative into the Safe Genes Project, as well as an IARPA initiative into protecting genes and turning them back to the baseline if they were ever accidentally or on purpose gene edited, we think that this has large-reaching economic and national security implications. The choice of the U.S. spy chief to call out gene editing as a potential weapon of mass destruction surprised some experts, and it was the only biotechnology appearing in its tally of 6 more conventional threats, like North Korea's suspected nuclear detonation, Syria's undeclared chemical weapons, and then the new Russian cruise missiles that might violate an international treaty. So, guys, we have all this crazy mad science research going on. We find out that they can use a common respiratory virus that is infected with CRISPR to get it inside of the human body to then splice and dice at will. Well, we started covering a story last week that I never finished, and I want to go back there. It's from MIT Technology Review under a section titled Rewriting Life, 5 ways to get CRISPR into the body. Number 1 would be gels and creams. There was a trial planned in China for treating human papillomavirus, and we read about that, and they're going to apply this gel directly to the cervix to help cure women with HPV infections. Number 2 is drinkable or edible CRISPR, and that would be the CRISPR pill or a drinkable liquid that would be able to then get edible CRISPR into your gut to be able to edit your gut bacteria, make fatal cuts to its own DNA, and kill potentially antibiotic-resistant bacteria. But number 3 is ear injectables, which I thought was kind of strange. Zhang Yichen, an ear specialist at Harvard University, again, is interested in whether CRISPR could reverse gradual hearing loss. Dedicated hair cells in the inner ear are what pick up sound and allow us to hear, and these cells can become damaged by too much exposure to loud noises or a flaw in a person's DNA. The latter is what Chen is hoping to treat with CRISPR. He has given mice direct injections of CRISPR into their ears to disable a gene called TCM1. All right, guys, make sure you have an awesome one.